Hello, everyone. I would like to start by thanking Elena and the organizer for giving me the opportunity to talk here today. Even if I'm not there in person, I'm really pleased to be able to share my work with you. My name is Alessandra Granata and I'm part of the Cambridge Stroke Research Group in the UK. This group brings together many different kinds of expertise. We have clinicians who see patients, imaging specialists, geneticists, bioinformaticians, and I lead the translational arm of the group, which means I use stem cell-based laboratory model to understand disease mechanism and to explore potential therapeutic approaches. The focus on my work is on small blood vessels in the brain. And these vessels are surrounded by two types of cells called smooth muscle cells on the arteriole and pericyte on the capillaries. And these are important cells that help the vessel keep its shape, control blood flow, and maintain a healthy communication with the surrounding brain tissue. Importantly, both smooth muscle cells and pericyte express the Notch3 protein, and we know this protein is affected by mutation in CADASIL. So these are crucial cells to understand when we study CADASIL. The small brain vessel form part of what we call the blood-brain barrier, or BBB. This is a very important barrier that protects the brain by controlling what goes in and out. And this is made by different cells type. They include smooth muscle cells and pericyte I mentioned earlier, brain endothelial cells, they line the inside of the vessel, and astrocyte from the brain. And there is another important component, extracellular matrix, or ECM. This is act as a scaffold that holds everything together and allow the cells to communicate with each other. And we know that Notch3 protein is very important to maintain a healthy extracellular matrix. However, in CADASIL, when Notch3 is mutated, the protein accumulates outside the cells in these clumps known as GOM, and this might disrupt the extracellular matrix, and in turn, this can weaken the blood-brain barrier, contributing to the disease progression. So the aim of my research is to develop precise, robust laboratory model to understand how the blood-brain barrier becomes damaged in CADASIL. And using this model, we hope to identify and test new drugs that can improve vascular health and can reduce disease-related changes. To do that, we use a very powerful tools in our laboratory called human-induced pluripotent stem cells, or iPSC technology. And I'll explain how it works. So we start with a very small skin biopsy from patient, and these skin cells are reprogrammed into iPSC. These, which are type of stem cells, can become any cells in the body. From the iPSC, now we can generate then the cells that we know are damaged in CADASILs, which are the vascular smooth muscle cells and pericyte. Now we have generated patient-derived cells in our laboratory that we can use to study the disease progression and to test new treatment. And this is very important for us. So I'll give you here an example of our patient-derived iPSC that we use to model CADASIL. The top images are healthy derived smooth muscle cells and CADASIL smooth muscle cells, which have been stained with marker that show their structure, respectively, activin and tubulin in green and red. And you can see the CADASIL cells look bigger, more spread out, and have a remarkable different appearance from the healthy cells. In the bottom image, the smooth muscle cells are stained for Notch3 here in red, and we can see in CADASIL cells, Notch3 accumulates outside the cells and form this bright cluster that we don't see in the healthy cells. And this is a very important observation because it showed that our model reproduce key feature of the disease that we see in patients. So we know that Notch3 accumulation is harmful and contribute to the disease. So the next question is, can we help the cells to clear this accumulation? How we do that? So in the cells, there is one pathway that normally remove excess and damaged protein, which is called autophagy, and is made of two components, is autophagosome and lysosome. They fuse together to degrade the protein. And what we see in CADASIL-derived smooth muscle cells, the two important autophagy-related protein, Beckley and LAMP2, they are increased. And this is indicative of autophagy pathway being disrupted or being going under stress in CADASIL. So this gives us a potential target. If we can support or correct autophagy, we might be able to reduce Notch3 buildup in these cells. So the next step we want to take is to test existing drugs that we know can control, influence autophagy. We have select 3 drugs that are safe and approved, and we want to treat our cells, CADASIL patient-derived cells, with these drugs and measure whether Notch3 accumulation is reduced and whether these cells regain a healthier behavior. If these treatment is successful, this approach could give future to therapeutic development for CADASIL. And with this, I would like to thank you all for listening. I would like to thank my team, our collaborator, and especially our colleagues in Spain who are working together with us on the autophagy project, and of course, the patient, the family who participate in this research. This work will not be possible without all of you. Thank you.